Alright, so for vlog number 2, let's try to define what synthetic biology is. And this is a very controversial topic because of how many people work in the synthetic biology field from different disciplines. And the result of all this is that they all bring their own flavors of the original definition of synthetic biology. And they all debate that their flavor is the definition of synthetic biology. And that's kind of really annoying because at the international level, it's very hard to define a unifying understanding of what synthetic biology is. And that makes it really hard to develop policies around this field. And that's really important because synthetic biology, you can do things like create bioweapons, and right now there's no real set guideline or standard to kind of control that. And so what we're going to try to do is stray away from these different flavors and just think about the field a bit more philosophically. So if you think about the field, it's synthetic biology. Biology is the study of life, and life is a very complicated, chaotic kind of thing. Because cells operate in a way where it's very hard to predict what's going to happen next with the cell, just based on a few parameters. So what synthetic biologists strive to do in general is to make biology more amenable to engineering. And to do this, you kind of have to make things a little bit more modular. And so what we have to think about is the central dogma. The central dogma is a flow of information from the DNA transcribed to RNA translated to protein. And proteins are, for the most part, the major agents of action within the cell. They do the things. They provide structure and they perform enzymatic reactions. They do all sorts of things. And so what synthetic biologists are looking into is the genetic structure of these cells. Can we modify the genetic structure? Can we enhance it by giving it new genes with new traits? Stuff like that. And what I'm trying to get at is something that's summarized very well by a quote from Richard Feynman, who is a very famous physicist. What I cannot create, I do not understand. So the idea is this. Hypothesis generation, creating theories, previously was very driven by curiosity. Why solve the structure of DNA? It doesn't have an immediate application when you think about it, but it has implications in the whole entire field of genetics. If you know what DNA looks like, then at least now you are able to, first of all, prove that DNA exists, and then prove that DNA is the stuff of inheritance. And those were the follow-up studies after Watson and Crick solved the structure of DNA, along with Rosalind Franklin's mostly important contribution. That's another topic. But that was kind of the idea of hypothesis generation back then. Nowadays, hypothesis generation is driven by finding patterns in large data sets. This is bioinformatics for the most part. And to understand how certain genetic components work based on how they work in an overall system. This is synthetic biology. And let's dive into that kind of meaning a bit more. Okay, so say you want to create some sort of GMO that is able to produce a pharmaceutical compound of interest. And to do this, what you're going to have to do is introduce DNA, plasmids, into, say, E. coli or yeast. And this DNA will encode for proteins that will participate in some kind of metabolic pathway that converts one chemical compound all the way to the pharmaceutically important one. And so to do this, you're going to have to find genes that encode for enzymes that can't participate in these steps. And you may find enzymes that have already been characterized before, so you know the function of it. It will convert this into this. But there might be steps where you find enzymes that aren't really the most well characterized, and so you're not exactly sure whether it's going to participate in that step or not. Okay? So you can take a classical reductionist approach where you purify this potential putative protein that might participate in this step, and then you study the hell out of it and learn all its different parameters, binding affinity, degradation rate, its structure, etc. And that might help you pinpoint exactly whether it's going to work or not. Or you can just try it out and put it into the system. And this is kind of the style of synthetic biology, where now we're going to generate a hypothesis of protein X by testing it out in a system where it has to participate in a pathway. And if this pathway is successful, you produce some of the pharmaceutical compound, then yeah, maybe protein X does participate in this pathway by doing this particular enzymatic function. And that's a very different way than just analyzing the hell out of it, you know. By doing this, you're able to kind of generate new hypotheses about protein X based on whether it functions in the system or not. And so this kind of brings us to a more unifying definition of what synthetic biology is. And I think this definition is unbiased and accurate, and it can be shared amongst the different disciplines that work in synthetic biology. So synthetic biology is the creation of non-natural biological systems by manipulating genes and genomes. And this is done so that we can design organisms to behave and function in a way that's modular and predictable, therefore amenable to engineering. And so by creating, synthetic biologists are able to develop new theories and analyze old theories in ways beyond the classical reductionist approach that has been used for the most part of the history of science. Alright folks, so that's a wrap for vlog number 2. It took me a good 50 tries to make this damn video, but as you noticed, I made this video multi-segmented because explaining synthetic biology was pretty challenging to do, even though I know it by heart, but it's still hard to explain it on camera. So if you haven't subscribed, please consider subscribing if you want to keep learning more about synthetic biology in Glasgow and Toronto. I upload twice a week, once a week. Thanks a lot.